Hi everyone. This is a presentation requested by the University of Bristol as part of my third year individual project. In this video, I'll be explaining the topic and the aim of my project, as well as give a clear insight into my progress so far. And finally, I'll give a quick overview of what I intend to do in the remaining period to meet my targets. First of all, we might have all asked ourselves this question at some point. How does an animal or a human being know where it is, and how does it exactly remember distant goals and then navigate efficiently towards them while avoiding hazards and barriers? Over the past 4 decades, research has revealed that the hippocampus formed the main area of the brain dealing with spatial memory. Now, spatial memory in simple terms is the storage and the retrieval of information within the brain that is needed both to plan a route to a desired location and to remember where an object is. Now, as animals navigate the world, they store information about the surroundings to form a coherent spatial representation of their environment in memory. As such, the functional roles of these neurons in and around the hippocampus of a freely moving rat have been characterized by their spatial firing patterns. In other words, as the rat explores its environment, neurons in the hippocampus increase their firing rate at specific locations. These so-called place cells increase their firing whenever a rat enters a preferred firing location or what is known as a place field. The firing of multiple place cells within the hippocampus can map an entire environment and encode crucial spatial information. Based on observation of place cell activity, it is possible to accurately decode an animal's location from given neural activity, and extensive efforts have been put into various decoding methods. However, for mathematical simplicity, most of these models neglect an important factor, and that is neurons are correlated. This project, therefore, aims to compare two statistical models of decoding from neural spikings, where one method has the assumption that neurons are independent and the other incorporates pairwise correlation. Once these are analyzed and compared, I'll be able to see really how including the correlation might improve the accuracy of the decoding. So what I've done so far is adopt a famous probabilistic approach for the decoding that is based on Bayesian probability. Given neural activity data, which by the way was obtained from Professor Matt Jones' labs here in Bristol, this approach aims to find the probability distribution of the rat's location at a time t, given a known count of spikes fired within a time interval, as it can be seen here in formula number 1. Now, the main aim behind using this approach is that in addition to assuming spikes having Poisson distribution, it also assumes that place cells across the hippocampus are independent. The outcome of this reconstruction method will then be compared to that of the next method. It is worth noting, however, that instead of reconstructing or decoding an individual position in this method, the computing results will be a distribution of positions whose peak correspond to the most likely reconstructed location of the animal, as shown in the bottom equation. So what I intend to do next in the upcoming weeks is to model another probabilistic approach of a different nature, and this will be the graphical Ising model. The Ising model is an important model in statistical physics that assumes binary variables and only local pairwise interactions between nodes, or for this context, interactions between place cells. Modeling this method and obtaining results from it, I'll be able to measure the accuracy of this decoder and establish a comparison between the two chosen decoders. And this will hopefully answer the question of whether or not considering the pairwise correlation between pairs of place cells will really have a direct effect on the accuracy of the decoder and conclusions can be drawn from there. This is the end of my video presentation. Thank you very much for watching.